very nice, very colorful photographs, and it's creating enjoyment of nature, and it creates some creativity, because for some people it will give them encouragement to create their own photos, or maybe surprise, or the structure of these beetles will create the interest to study it, or for example the surprise of these hairs of these beetles. Actually, a good initiation to study the surface of these beetles under the scanning microscope in a time of a university or after university. And of course, in using these books, you can see how many different insects collected in collections, in collections, in the museums, and for scientific collections, for scientific purposes, usually making collections for study morphology, taxonomy, systematics, and study also physiology, to study behavior of insects, how we're creating the huge populations. This is very important. And this can be useful very much, for instance, in agricultural entomology, in creation of different methods, how to control some insects who are becoming dangerous pests for agricultural stocks. Oh, like this huge longhorn beetles, some of them really huge and very attractive for nice private or governmental collection. Or some of them very interesting to study their life cycle inside rotten wood, and to find some of the parasitoids from Hymenoptera, or to study different morphological features like eyes, like some setae, like some legs, like some sensilla on legs, can be very, very important for the development of biomorphology, biomimetics. Biomimetics is called how to use different adaptations which is used in insects and other animals for human beings. And studying some books, you can recognize how important to use this information for bioinformatics, how it's possible to keep now all this information as digitalized information, digitalized photos, not only on printed in books, but also in files in some databases, in computer databases, and this information can be freely collected and distributed for people. How quickly you can show discovery, you can find some discovery of other people in behavior, in morphology, in trophic relations between insects and other insects and in other vertebrates, or trophic relations with the plants, because many insects were developing like phytophagous insects, were feeding on many plants, which are our domestic plants, or they can be very dangerous for domestic plants, or we can destroy some weeds, and in that situation some insects can make biological control of weeds or some dangerous plants in farm fields. And development of insects from egg to larvae to pupa is really huge subject for this study. This is physiology and morphology changes, and this process of metamorphosis just still keeping a lot of unstudied features, unstudied material, because it's difficult to study this process of metamorphosis on different groups of insects. Some groups this is even unstudied, and how to control it, how to manipulate it, how to observe it on healthy pupae, larvae, eggs, and adults. It's very important physiological study. And even study of some insects inside amber, here white and black, but originally these insects were in amber. If you are just lucky to have small pieces of amber, you are very lucky to have it, because it can be useful for the study by scientists. But here you see some of the insects alive. Very important to use photographs of live insects, because they show their natural behavior, which is opening the life cycle, lifestyle, showing trophic relations, and how to study it. There are different methods for studying insects, definitely. As I showed you some photographs, very important to take photos of insects. And now we can go to methods of study in insects. For example, we can use small video camera to catch some behavioral features. You can use this camera like this. Just don't break it, don't miss it, don't destroy it, to catch some very, very important, very interesting moments of behavior of these insects. And with this current style of technology, current level of technology, it's becoming easier and easier, especially with a huge big zoom. You can use it to manipulate, to zoom tiny insect, and insect will be big, and you can catch very tiny, unstudied before moments, details of biology of phytophagous insects, or the parasitoids like these tiny parasitoids, unstudied, completely unstudied. And it's very important to study them, because, for example, as I said, these tiny beans you can collect at home, but some beans very widely distributed in tropical, semi-tropical areas in such long, short pods, and some bean beetles laying eggs on pods, and many entomophagous insects, which are just on this picture, just behind of me, some entomophagous insects from the order Hymenoptera attacking these beetles. These tiny Hymenoptera parasitoids, tiny from 0.5 millimeter, half of millimeter, up to 1 millimeter only, they attack larvae or attack eggs of bean beetles, just on beans or just on pods, just on plant. And if you collect plant with beans, if you collect eggs of plants in tiny jars, like in petri dishes, in some plastic tubes, in some small containers, you can collect phytophagous insect, and you can breed parasitoids, egg parasitoids, larval parasitoids, egg larval, and pupal parasitoids, and sometimes even adult parasitoids. They're all available in the world of parasitic Hymenoptera insects. And some of these insects can be new for science. Yes, some of insects collected, rear it carefully in small jars, can be new insects, new for science insects. What means rear it? If you are making some samples where you are keeping eggs, larvae of insects, like in these jars, from eggs, from larvae, from pupae of insects, can be these beautiful parasitoids rear-it, and if you fix them, if you keep them and just dry or in alcohol, specialists like me or other entomologists can identify the scientific name of this parasitic insect. And to know name of parasitic insect, like name of these all tiny small parasitoids, is very important to know exact knowledge about biodiversity of parasitic insects or biodiversity of Hymenoptera in the nature. And entomologists, scientists, on the basis of this material can publish exact scientific papers with indication of exact scientific names of this or that insect or parasitoid. You can watch on my channel how to breed, how to keep insects in different cages, like that from these cages, and how to breed them. So this is a technical entomology. You can keep insects, you don't need even kill them immediately, all of them, but you can keep them in jars like this, from a milk, from a cream, from these tiny plastic jars, from cheese. You can make here holes, put here some plants or some wood and keep it. Or with this, for example, from cheese, polished cheese with flattened, with small cover, easy to put here a sample with plants, with some small tiny pieces of plants, with leaves, with wood, close it, and you can observe here through transparent plastic as well, from transparent plastic. And this observation can be very useful to make collection of insects for the scientific study. So the next part, after observation in the nature, where you can take photos, where you can take videos, where you can record very special moment of biology of this or that insect. It's very important to take some of insects at least, not all, maybe not hundreds, but some of insects for scientific collection. Fix them, because, as I said, very important to make very correct identification of name of insect. Sometimes with just a photo, it's not enough to make correct identification. Scientists need to watch under the microscope tiny insects, because, for example, this insect very tiny, just 1 millimeter and less. Very important to have tiny samples with a tiny inside plastic tubes like that, with cotton, closed with cotton or closed with plastic, and if it is, will be with alcohol, so it can be dry like that, or in alcohol in this tiny jars like that, tubes, different kind of tubes, or this can be tube with a cap, so it is closed carefully, very carefully, and put it inside another glass jar. Especially very important to have a sample of insect and also the label, piece of paper which is named label, with indication of a country, city, village, name of collector, plant of collection, where it was collected, and date of collecting. So in this case, these tiny small pieces will be very valuable. These small tubes with insects inside, with indication of all data on the label, will be very useful, very important scientific material for the study. There are a lot of different methods for collecting, as I showed in my previous video before. For example, I have some samples collected in alcohol. using different plates. So these plates are called yellow pen traps. Plates. These are tiny plates. We're filling these plates with water and with some drop of shampoo, and so they are called murky traps or yellow pen traps. Easy to remember. So that if they're deeper, they're better. And so water, shampoo, and just 1 spoon, big spoon of salt, common salt, natrium flour for 1 big bottle of 2 liters or 1.5 liter, 1 spoon of salt for fixation of insects. So you fill water with shampoo here and salt inside these plastic plates and put them just on soil, on stones near the river, near the lake, near the pond. Sometimes it's even possible to attach it to the tree, to a stem of tree, even put on the level of 1 meter, 2 meters, or even 3 meters and higher on the tree, together with the water with shampoo, and insects are attracted by the yellow color. It's already proved by different studies that yellow color is the best. Sometimes not green, not white. Sometimes blue color is useful, but yellow is the best color. So insects will be flying, crawling, jumping, and all of them will just go through the surface of water with shampoo and dying, sorry about that, inside water with shampoo. And after that, you can use just a very tiny, very small aquarium net to collect all the insects together. All insects, you can collect them in a glass jars. Wash these insects in a water in laboratory, or just wash with fresh water without shampoo. Clean them from shampoo and then fix them again. Finally in 70 or 96% ethanol alcohol for the next scientific study. And how it can be looked like. For example, if you collect it, this can be, if they dry, so they die. So you see here, this insect probably destroyed because alcohol evaporated. But if insects collected properly and put here inside alcohol, they will be saved here for several years, for many years. If we put insects in a freezer or in a refrigerator, these insects will be saved for a long time, and some of them, when I already selected some from big box into the small box, and here I'm wrote the label, material was selected, because I need only small insects from this big jar with different insects. So I selected the best, which are most important for me, special taxonomical group. So this is a method of collecting of insects in yellow pen traps and then put them in alcohol for following next taxonomical study and identification of insects. As I said, to know correct name of different groups of insects which were collected all together, because inside yellow pen trap you can collect many different families, orders, groups of insects, species. So I select only 1 and 2 or 3 separate families of Hymenoptera insects, of small calcid wasps, which are here. My favorite insects are here. Egg parasitoids, Trichogramma and Trichogrammatida family. Egg parasitoids of beetles and butterflies. If you can collect eggs of beetles or butterflies and keep them in jars, tiny small eggs of beetles and butterflies, maybe some of cicadas, keep them inside jars, and after that you can look carefully inside jars, and you can find the tiny small insects hatched, emerged from eggs of their hosts. So this will be the most valuable material for my study, and I will be very pleased to share my knowledge with you to make identification for you species of Trichogramma and other genera of Trichogrammatidae family, or another egg parasitoids of calcid wasps, superfamily Calcidoideae. Of course, for many different groups of insects, you still can use butterfly net. For Hymenopterus, people use the name sweeping net because we are sweeping, just making some movements like sweeping. But some people do not understand what means sweeping net. Easier to say butterfly net. Butterfly net can be short size, or sometimes it can be dismounted and it can be much bigger. It's becoming much big, and this is still not very big one. Some Japanese entomologists they use, and some entomologists who are studying in butterflies in tropics, we use very long stick, to move very fast, very quickly, to catch some morpho, some very tiny butterflies, very quickly. So because this is a butterfly net, also can be useful for collecting different groups of insects. And as I said already, for creation of a scientific collection. And finally, I show you a little bit of collection, because some people like to create just exposition of collection like this. So they collect some colorful butterflies, colorful beetles, or some people they just buy them on auctions in internet. But others using different methods of collecting for creation of it. The amateurs or scientific collections, like with this crocodile, this is exposition for educational collection. I wouldn't say this is scientific collection, more educational for exposition to show different groups of insects. Like here some beetles, scarabid beetles, some this is Blattodea, and here some scarabid beetles, and here some weevils from Curculianidae. And very important, I forgot to say, making this collection, don't forget about this important stuff. If you are making collection, if you are making observation, don't forget you need to have observation diary of your observations. This is very important, where you can write date and your observation, where you indicate temperature, plants, place of collecting, because later you will forget it, all small tiny details, and peculiarities of behavior which you just observed with different insects. Collecting boxes can be big size, and this is my maybe first wooden box. This is from art box used for painters. So here some bees, from my old collection, which I collected just in university. Some bees still here with labels. I'm sorted them and put in different places, but it was used actually originally for art painting, for different colors and painting here. So this is such a big box, not for exposition, but must be closed very toughly, very carefully, without any crevices. Or you must use maybe here scotch to cover it and for protection against insects who can eat it inside. but this is a separated video. So this is exposition, for example, with different bees, hornets, bumblebees, yes, here small bumblebees, wild bees, especially you need to indicate to have some labels and different groups. This is also exposition collection. And educational, this one, educational collection with hornets, or European, for example, we don't have Asian hornet, Vespa mandarina yet. We don't have a Vespa velutina yet, but when we will have Vespa velutina, I will create big collection like this with Vespa velutina, which is invasive species and which is going to invade different countries in Europe step by step, year by year, and sooner or later it will come to the east because of global warming of climate, and they're coming to different countries 100 or more kilometers every year. So here this is hornet nest and different workers and some big, even some huge, I would say, some huge queens of hornets. And here's some exposition about honeybees and some hornets as well. So here's some workers, some queens, some drones, some wasps, German wasps, some big hornets, and here some pieces of combs and queen cells, and here for comparison some wild bees of Andronidae family. So, for example, this is a small piece of my educational collection, which but this is only a small piece. Usually, people have many drawers, many boxes, and of course, finally, I show you admiration of insects when people keeping different insects as a living stock. Living stock, it means if you like insects and you have a possibility to keep insects in house or just in laboratory for their study. For example, here I have a piece of wood, and this is a piece of wood with the Zaphobos. This is a tiny larvae, not tiny larvae, very big larvae. I say tiny. Some people worry about it, but they are not dangerous. I showed them in my video. I showed how to breed them, how to keep it this stock, about peculiarities of biology, about peculiarities how to feed them with a different food, how to keep them safe. And you see here, they're safely sitting here and eating just this and just biting, not biting. They have no sting, no bite. They're eating this rotten wood. Old wood, actually not rotten. And of course, some people need to keep this technical collection for the study, or maybe to use these insects for like food for another animals, another small vertebrates, or invertebrates like spiders. I don't have too much spiders in my collection because mostly I have insects. Like here, finally I show you small, as I said about Zaphobos. So we're sitting here, some of these big beetle, Zaphobos beetles. From time to time I'm telling you the story about these beetles, how to keep their culture. So this was a story about different type of collections, different type of admiration of insects, and when it was started usually for amateurs and entomologists. So I'm very pleased that you came to this video stream. Of course, it was recorded and it will be repeated on my webpage on my YouTube channel. And write in your comments which insects are you keeping on yourself, which insects are the most interesting for you, which groups of insects is the most interesting for you. Lepidoptera, butterflies and moths, beetles, grasshoppers, cockroaches, larvae, maybe you aquatic beetles, maybe caterpillars, maybe you are just breeding caterpillars.